The lock and key model for enzymes is basically what it sounds like. It's that the enzyme itself is like a lock and the substrate itself is like a key. And so what you end up getting is this very specific substrate, one key that likes the enzyme, to break down our products into basically into two products. So it's really the shape and the chemical properties of that active site and the substrate that match each other, like a very specific puzzle. Now what we know today, and we still lend toward the lock and key model, but we also talk most about the induced fit model. And what the induced fit model is, it's basically this enzyme is not necessarily like a lock, but the enzyme itself is going to hug onto that substrate. And so you have some flexibility that you see within the enzyme, and it's all due to very weak chemical bonds between the substrate and the active site. And these bonds are going to basically put a lot of pressure onto our substrate, and so what that's going to do on those bonds is basically break the bonds, lower that activation energy, and cause you to have your chemical reaction. The other thing that we know about these enzymes is that they actually aren't very specific to only one type of substrate. And so they actually can catalyze several different reactions. An example of this would be something called a hexokinase. It's an enzyme that can actually help phosphorylate different hexoses, including something like a glucose. So glucose is a 6 ring, and so that's just one example of a hexose that a hexokinase can break down.